it unique? From that particular receptors, signals are transmitted to the different cellular systems of our body. And from the very beginning of the microorganism invasion, our body is cautious, is very much clear to our immune system that what is coming. So it's a unique concept and Nobel Prize winning concept of microbiology, the TLR, the toll-like receptor. The immunoglobulin, another soluble molecule of the acquired immune system. So the immune response is interrelated. The cell-mediated immunity and humoral immunity, they take part in immune response hand in hand, and they are interdependent. The immune response, if we consider it as the humoral portion where the different immunoglobulins are there, they produce the primary immune response whenever any antigen enters the body. The primary immune response is very immediate. Is it true? Yes, it is true. The primary immune response, the antigen enters and the body's response. Is it immediate? Is it immediate? No. The immune system takes long to recognize it. The initial phase is lag phase. What type of organism? They receive the signal from the TLR. Now the processing part of the antigen. The antigens are processed within the cell. And the processed antigen are expressed within our system by the different receptors. The affected cell, they flag out inside our peripheral blood system, but hematopoiesis inside the blood system. They flag out the peripheral blood. Sebas. Now, the immune cells, the always in surveillance, macrophage cells, dendritic cells, they receive the signal. And from there, they engulf them or process them. There may be opsonization. So many pathways are there, and they started producing different type of immune responses. Now, immune response may be protective, may be unresponsive, may be destructive too. So, the immune response, when elicited, is good for us, but when there is no response, this is called anergy. There is a compromise between the organism and the human system. In case of long parasitic infestation, the parasite surviving within the system and our immune system not producing anything against the parasite. So there is a compromise between parasite and our immune system. We will discuss all these things, where it happens, why there is compromisation, and what sort of immune response body produces. The first one is the humoral one, that is through the IgM and IgG. If you see, this picture, the first antigen, after the entry of the antigen, and this side is the concentration of antibody, and here is the primary antigen exposure. So this is the lag phase of at least 10 days. No response, please. There is no response. The processing stage of the antigen. After that, the first antibody to appear is IgM. IgM appears first. The stage is lag phase, log phase, and then exponential phase. Plateau is reached, and then the declining phase at the level of 16 to 18 days. IgM declines. IgG, just after 12 days or 13 days, start picking up. And now, it is the turn of IgG to protect throughout the period of at least 30 days. So, this is the primary immune response. But what happens when that same organism enters again? Say for example, the dengue, say for example, the COVID, second infection of COVID, or any sorts of infection, maybe amoeba, maybe parasite, whatever it may be. The second entry of the same organism having the same genotype, at least 95% homology. With the entry of that organism, the existing antibody titer falls because it reacts with the antigen. So, antigen-antibody complex form. That is why this is the negative phase. With the secondary antigen exposure, if you measure the antibody level, it goes down. Then once again, there is a peak, and this peak is after 4 to 5 days. The time period is less now in compared to primary immune response. The secondary immune response is more efficient. Not only IgM, but the IgG from the memory cells, it starts proliferating. There is clonal proliferation, and then IgG is persisting. So, IgG is protecting, and the secondary immune response is more efficient than the primary one. The memory cells that persist are proliferated. So, this immune response is humoral immune response, and humoral immune response is usually B cell mediated. Now, the cell mediated immune system. The humoral mediated immune system mostly acts on the extracellular organism. The extracellular present outside the cell. But what happens in case of intracellular organism? They are hiding themselves within the cell. This is the cell mediated immune response. They take the upper hand at that particular moment. So, this is the nature of antigen. What sort of antigen is that? Whether it is cell mediated immunity or humoral immunity. They have to decide first. Now, the cell mediated immunity, where antigen is taken up by the APC cell, antigen presenting cell. What are they? The dendritic cells, the macrophage cells. They are the antigen presenting cells. Neutrophils, they present the cell. Initially, they process. If the antigen is endogenous, in case of viral infection, the virus integrates within the genome of the host cells. This is an endogenous antigen. In case of bacteria, which are exogenous, not all, but these exogenous antigens, the pathway is different. So, the processing for endogenous antigen, they are processed in endoplasmic reticulum as well as on the human cells and also in the Golgi body, the antigen presenting cells of human, no? So, they process it and they are expressed outside by the MHC receptors, that is human leukocytic receptors present in all leukocytes, human leukocytic antigen HLA or MHC, major histocompatibility complex, they express it with this particular hand, that this is the processed antigen. So, endogenous antigen are expressed via MHC1. Endogenous antigens are processed and expressed via MHC1 or HLA1. Just the reverse in case of exogenous antigen. They are processed via and expressed via MHC2. But the processing is easy. Simply they receive the antigen and express it. No complex, no processing, nothing. So, the processing of the antigen that takes place within the APC cell, and after the processed antigen into different peptides, smaller peptides, they are expressed via MHC1 and MHC2. This is the antigen presenting pathway. Now, our immune cells, to receive, to acknowledge the particular MHC or particular MHC1 or MHC2, they must learn to recognize them. This one is presented by MHC and this one is presented by MHC2. So, learning this phase, how to recognize them, there is a training center inside our body. That training center is thymus, and thymus. So, inside the thymus, all the cells, especially the T cells, they are named because of that reason that they are processed in the thymus. That is why they are T cells. B cell, they are processed in the bone marrow. That is why they are B cell. So, T cells, hey, AC ta chali dao ekta duto. So, T cell, they are processed in the thymus. This processing is unique. The primordial cells in the embryonic life, they start from the yolk sac, from the bursa of fabricus, the mammalian counterpart is bone marrow, and after production of these, they migrate to the thymus. In the thymus, they are first exposed to the self antigen, that is MHC1 and MHC2. If they can recognize it, yes, this is MHC1, this is the self antigen, or MHC2, then only they are selected. Then only they are selected, otherwise they are deleted out. They are referred to the programmed cell death or apoptosis. So, the selection, this particular selection of MHC1 and MHC2 recognition is known as positive selection. Those positive cells who can recognize are selected. After that, the same T cells are presented to thymic epithelium on the thymus. On the thymic epithelium, the self antigens are there, and if the reaction is very aggressive against the self antigen of thymus, they are deleted out. So, the same reactions, when the same cells against the self antigen on the thymic epithelium, they show the aggressive behavior or they are hyper responsive, they are deleted out. So, this is negative selection. Is it clear? Is it clear, students? MSC1 and MSC2, recognition, positive selection, and hyper-responsive against the thymic epithelium, the different self-antigens, hyper-responsive, they are deleted, this is negative selection. Here, all these cells are DIN, they are doubly positive, having both CD4 and CD8 molecules on their surface. These are known as cluster differentiating cells, cluster differentiation. That is, on the surface of the cells, different clusters are presented. Here the cells are DIN cell or doubly positive cell. Both CD4 and CD8 receptors are there, doubly positive within the thymus. Now, with the different hormone and enzymatic action on thymus, they are differentiated out to CD4 and CD8 cells. Having only the CD4 receptors at CD4, having only the CD8 receptors at CD8, they are now differentiated. These receptors, the transmembrane receptors expressed outside the cell, they are the cluster differentiating cells. They are a particular type of clusters of CD4 and particular type of clusters of CD8. So, the different type of cluster differentiating cells are there, and these are two of the most important examples are CD4 and CD8 cells. Now, with this CD4 and CD8, the CD4, they can only recognize MHC2, MHC2, that is HLA 2, and the CD8 can only recognize MHC1 and HLA1. These are same, HLA, human leukocytic antigen or major histocompatibility complex cells. So, these are the self-antigen. CD4 can recognize the self-antigen of MHC2 and CD8 can recognize the self-antigen of MHC1. So, are differentiated out. So, our soldiers are ready on thymus now. Now, they move to the peripheral lymphoid system. They are ready for fighting. Any antigen, whether it is exogenous, whether it is endogenous or exogenous. Now, we are ready with the CD4 and CD8 arm. Now, the question is the T cell and B cell maturity. So, this is the process of T cell maturity that happens within the thymus. These cells, T cells, are further differentiated into different clones. But for we people, the CD8 and CD4 is very important short note. The B cell, on the other hand, they are formed within the bone marrow. Within the bone marrow, and selectively within the bone marrow, the first immunoglobulin appear is IgM. First immunoglobulin appear is IgM. Subsequently, on different antigenic challenge in the bone marrow, from IgD to IgE, IgA and IgG, these are formed. This particular behavior of changing one immunoglobulin to another immunoglobulin is known as class switching. Within the B cell, particular genome structures are there, VDJC. the variable, diverse, junctional and constant regions. There is a hypermutation or genetic reassortments within the genome, and that is specific against a particular antigen. When parasitic antigens are there, IgE will be produced, IgM followed by IgE. When IgG is required, after the IgM, IgG will be produced. So there is a genomic reassortment within the structure, within the B cell and against the different antigens. So this is the B cell differentiation process. And the B cell, moreover, they have with the immunoglobulin attached on the surface, they have the similar BCR receptors, B cell receptors, and this is also immunoglobulin-like, and they have Fc receptors. The Fc receptors, you know that every immunoglobulin there is Fc portion, and to bind that immunoglobulins there is the receptor on the cell surface of B, that is Fc receptors. Fc receptor site is there. And another receptor site is erythrocytic antibody complement receptor site is there on the surface of B cell. But in contrary, in T cell, there is TCR. It is of the same immunoglobulin family, almost similar this TCR. This TCR having alpha and beta chain on either side, alpha and beta, mostly in case of 80% cases, but maybe gamma delta chain in 20% cases, there is possibility of gamma delta chain. So mostly there is alpha beta chain of TCR receptor. CD3, a cluster differentiating cell to identify any antigen within the group of the TCR. They are the signal transmitting cluster. They transmit the signal inside. So that's it, that is CD3, that is present within the outside the T cell. And interleukin 2 receptors is there, and one more is SRBC, that is sheep RBC recognition receptor outside the T cell. These two are the two different pictures. That is the difference between the T cell and B cell. T cell and B cell. So the binding receptors are different. Now, the role of thymus and bone marrow. Now we know what is the role of bone marrow and thymus. Difference between T cell and B cell. MHC1 and MHC2, the leukocytic antigen. And now cytokines, interleukin and subsets of T cells, and the central process of immune response. So when the antigens whether it is exogenous or endogenous, they are presented and they are processed by the APC cell. The APC cell, by their receptor, they express it, and the processed antigen, say for this is exogenous antigen, then the receptor will be MHC 1 or 2, exogenous 2, MHC 2, and in case of endogenous it is MHC 1, isn't it? So by the MHC 2 they are presenting that this is the antigen, this is the processed antigen. Now the CD4 cell to recognize, CD4 cell will recognize MHC2. Any molecule expressed via the MHC2 will be recognized by CD4 cells. So this is a CD4 cell. So CD4 cell within the group, the antigen is there. Now as soon as with this receptor TCR within the group, the antigen is there. CD3 send the signal inside. With the signal, different hormone-like materials are secreted. These are interleukins. These are known as cytokines. So, these are IL-2 which is secreted from the Th cell. This cell is CD4 is also known as Th0 cell because it is Th0 because initially it was not exposed to any antigen. Now, after the antigenic exposure, this particular clone with the help of IL-2 also act on the IL-2 receptor of the same cells and also can act on different cells. So, this is a hormone-like material, cytokine. So, with this action, they are clonally proliferated, Th0 cell. Now, it proliferates to Th1 and Th2. The same Th0 cell proliferated into Th1 and Th2. The Th1 mostly with the cell mediated immunity and Th2 with the HMI. Th1 where interleukin 2 and interleukin 12 they play the major role, and the macrophage cell is stimulated. With the stimulation of macrophage cells, nitrous oxide like material or superoxide dismuted like material, highly toxic material are secreted and secreted on the microorganisms. Those are intracellular. So, all the intracellular microorganisms, they started secreting the nitrous oxide like material or superoxide dismutase, highly toxic materials. So, there is the lysis of the affected cell. This pathway is cell mediated immunity. You can understand that the antigen is present in is exogenous antigen. So, the pathway through the exogenous antigen is APC cell processed antigen to CD4 cell. As soon as CD4 cell receive the signal, CD3 process it and send the signal to inside the different genes of the CD4, and IL-2 is secreted out. IL-2 not only activates the cell, but it has different role to play. So, it activates the Th0 cell or CD4 cell, and now the CD4 cell is clonally proliferated to Th1 and Th2. The Th1 under the influence of interleukin 2 and interleukin 12 signal to macrophage cell, and macrophage cell secrete nitrous oxide like substances, toxic materials, and the intracellular materials within the cell is destroyed or lysis of the cell. This procedure is cell mediated immunity. Is it clear? Similarly, if the antigen is endogenous, it will be presented by MHC1, and MHC1, through the MHC1, the signal will be received by CD8 cell, not the CD4 cell. That will be received by CD8 cell. And similarly, CD8 cell will destroy the intracellular organism as well as extracellular organism. But here, when the exogenous antigen appears, not only the Th1 is stimulated, but as the Th0 cell is stimulated, there is also to some extent stimulation of Th2. Th2 under the influence of interleukin 4, 5 and 6 they stimulate eosinophil, and eosinophil, as soon as eosinophils are stimulated, they started secreting some toxic materials, granzymes-like substance, and they also participate with the intracellular killing. So, both are practically working hand in hand. But interleukin 10 and interleukin 10 and 12, they are to control the immune response, which will take the upper hand. Is it Th1 or Th2? But to some extent, they goes hand in hand and both the response are produced. कुछ आया समझ में? नहीं, गड़बड़ हो गया, बुझा गया? Shall I repeat? আবার বলবো, বেশ। এক্সোজেনাস অ্যান্টিজেন এন্ড এন্ডোজেনাস অ্যান্টিজেন, এটা প্রসেসড হলো, এখানে প্রসেসড হলো অ্যান্টিজেন, প্রসেসড অ্যান্টিজেন প্রেজেন্টেড হলো বাইরে। এই প্রসেসড অ্যান্টিজেন প্রেজেন্টেড হলো CD4 সেল এক্সোজেনাস অ্যান্টিজেনের ক্ষেত্রে। যখন MHC1 দিয়ে প্রেজেন্টেড হয়েছে তখন CD8 সেল, আর যখন MHC2 দিয়ে প্রেজেন্টেড হয়েছে তখন CD4 সেল তাকে রিকগনাইজ করলো। ওরা অ্যান্টিজেনকে রিকগনাইজ করে না, ওরা করে MHC1 আর MHC2 কে রিকগনাইজ করে। কাকে দিয়ে প্রেজেন্টেড হয়েছে তাকে ধরে। ইজ ইট ক্লিয়ার? তাহলে সেই MHC2 দিয়ে যখন প্রেজেন্টেড হয়েছে তখন MHC2 কে ও ধরলো। যখন এই গ্রুপের মধ্যে এই সিগন্যালটা এলো, সঙ্গে সঙ্গে CD3 সেলস সেই সিগন্যালটাকে প্রসেস করে ভেতরে পাঠিয়ে দিল। তাতে কি হলো? তাতে এই Th0 সেল যে ইনঅ্যাক্টিভ ছিল, নেইভ ছিল, যে আগে কখনো অ্যান্টিজেন এক্সপোজার হয়নি, সে অ্যান্টিজেন এক্সপোজারের সঙ্গে সঙ্গে ক্লোনালি প্রলিফারেটেড হয়ে গেল Th1 আর Th2 সেলে। Th1 সেলটা মূলত তখন অ্যাক্টিভ হয় যখন ইন্ট্রাসেলুলার কিলিং এর দরকার হয়। তবে সঙ্গে সঙ্গে কি আর একটু আধটু Th2 স্টিমুলেটেড হয় না? তাও হতে পারে। Th1 যখন ইন্টারলিউকিন 2 আর ইন্টারলিউকিন 12 এসে Th1 এর উপর অ্যাকশন করলো, তখন এ কি ম্যাক্রোফাজ সেলকে অ্যাক্টিভ করলো। ম্যাক্রোফাজ সেলের ভেতরে যে সমস্ত অ্যান্টিজেন আছে ইন্ট্রাসেলুলার কিলিং হয়ে গেল। ম্যাক্রোফাজ সেল তো অ্যান্টিজেন নিয়ে এসেছিল, ওইখানে ম্যাক্রোফাজ সেল এনেছিল, সে তখন নাইট্রাস অক্সাইডের মধ্যে দিয়ে মারা গেল। তাহলে সেল লাইসিস হয়ে গেল। সেখানে খানিকটা পার্টিসিপেট এডিসিসিও করতে পারে। যেমন কোনো কারণে একটা ফ্যাগোলাইসোজোম তৈরি করে ফেললো ব্যাকটেরিয়া, তার মধ্যে সার্ভাইভ করে গেল। তখন এডিসিসি সেলকে ওডে বললো যে তুমিও স্টিমুলেটেড হয়ে আমার সঙ্গে এসো, হ্যান্ড ইন হ্যান্ড আমরা কাজ করি। তখন এ কি করলো? Th-sol, IL-4, IL-5, IL-6 এর মধ্যে দিয়ে ইওসিনোফিলকে স্টিমুলেট করলো, বা অ্যান্টিবডি ডিপেন্ডেন্ট সাইটোটক্সিক সেলকে, এডিসিসি সেলকে স্টিমুলেট করলো। ওই এডিসিসি সেল আর ইওসিনোফিল আরো বড় বড় দাদা, এরা গিয়ে গ্র্যানজাইমস বা নানা রকম এনজাইম সিক্রিয়েট করলো। তাহলে এর সঙ্গে টক্সিক ম্যাটেরিয়ালস আরো যোগ হলো। অনেক রকম টক্সিক ম্যাটেরিয়ালস এর মধ্যে দিয়ে তখন ইন্ট্রাসেলুলার কিলিং হয়ে গেল। বোঝা গেল এবার? বেশ, তাহলে এবারে now this immune response, this particular response, the primary stimulus is this antigen. এই অ্যান্টিজেনটা যখন প্রেজেন্টেড হলো MHC1 আর MHC2 দিয়ে, সেটা প্রাইমারি স্টিমুলাস। বাট প্রাইমারি স্টিমুলাস অ্যালোন cannot produce the immune response. It needs the secondary co-stimulatory effect. Co-stimulation, a secondary stimulation. What is that? Macrophage cell, it has B cell, B7, not the B cell, B7. A B7 cluster differentiating molecule, B7 molecule on the surface. Macrophage cell has B7 cluster differentiating molecule on the surface. They send a signal to the CD28 receptors on the CD4. CD4, it has another receptor known as CD28. And through this, the interleukin 1, another cytokine is secreted. As soon as the secondary co-stimulation comes, the clonal proliferation started to its best. So the clonal proliferation or Th0 to Th1, Th2 proliferation not only depends on the peptide molecule expressed by MHC2. The co-stimulation is required. If there is no co-stimulation, what will happen? No immune response. Then the status is energy. Compromise. ও ভাই অ্যান্টিজেন তুমি যে আছো আছো, আমার কিছু করার নেই। নো সেকেন্ডারি রেসপন্স, দুজনে মিলেমিশে থেকে গেল বহাল তবিয়াতে হচ্ছে। তো দিস co-stimulation is absolutely required. Until and unless there is co-stimulation, there will be no immune response. So don't forget to write this co-stimulatory effect of the APC cell, the primary stimulus and co-stimulus. বোঝা গেছে? এবার ছবিটা এঁকে নিয়েছো? বইয়ে নেই ভালো, অত্যন্ত বাজেভাবে লেখা, কিচ্ছু বোঝা যায় না। একেবারে ছবিটা দেখে মাথায় ঢুকিয়ে ফেলো, 3 ডাইমেনশনাল, একেবারে পরিষ্কার হয়ে যাবে। অত্যন্ত বাজে লেখা, বিভিন্ন রকম সেল ধরে ধরে লিখেছে, ইমিউন রেসপন্সটাই নেই। এটা হচ্ছে কমপ্লিট পিকচার। কোথাও অসুবিধা হলে বলো, আমি বলে দিচ্ছি। Products দুটো আর্ম আছে, humoral and cell mediated, provides protection to the host by secreting antibodies, primary defense against extracellular pathogen, recurrent viral attack, participate in immediate hypersensitivity reactions. এটা হচ্ছে humoral antibiotic, তাই না? hypersensitivity 1, 2, 3 তোমরা পড়েছো, ওগুলো humoral. No role against intracellular antigen, তার মানে humoral antibody, no role on intracellular killing. আর মোস্টলি ভাইরাস থেকে শুরু করে সব খতরনাক অর্গানিজমস, দে আর ইন্ট্রাসেলুলার। Cell-mediated immune response plays a crucial role providing protection against intracellular microbes as well as tumor cells protection এর ক্ষেত্রেও এটা কাজে দেয়। Mainly T-mediated, especially cytotoxic T cells. T সেলের অনেকগুলো সাবসেট আছে, কিছু কিছু যেমন আমি বললাম TH সেল একটা ক্লাসে তো আর হয় না, TH সেল TH1 TH2 যেমন আছে সেরকম T সাইটোটক্সিক সেল আছে। T সাইটোটক্সিক সেল একটা সাবসেট অফ সেলস, সেখানে সেটা সাইটোটক্সিসিটি করে শুধু। সেই সাইটোটক্সিসিটি নট অনলি সে শুধু টি ডিপেন্ডেন্ট হবে, টি ইন্ডিপেন্ডেন্টও হতে পারে, সরাসরি তার অ্যান্টিজেন ধরবার ক্ষমতা আছে। CMI, AMI, they are interdependent, সেটা তো আমরা ছবি দেখেই বুঝতে পারছি। হেল্পার টি সেলস, ওই টিএইচ সেলের নাম হেল্পার টি সেলস। টি সেল সাবসেট বলে একটা প্রশ্ন পরীক্ষায় আসে, সেখানে কি করে থাইমিক কন্ট্রোল থেকে টি সেল তৈরি হলো এবং ডিফারেন্ট ক্লাস্টার্স টি সেলের তৈরি হলো, সেগুলো লিখতে হয়। তো সেগুলো তোমাদের এখন লেখাটা কোনো অসুবিধার কিছু নেই, অ্যাটলিস্ট লিখতে পারবে। CD4 CD8 সেলগুলোই আমরা বেশি দেখি, আর টি সাপ্রেসার সেলস আর টি ডিটিএইচ সেলস এগুলো আমরা দেখি, বইয়ে এক লাইন আছে, দেখে নিলেই হয়ে যাবে। মোস্টলি এই দুটোই ঝামেলার, এগুলোই পড়া হয়ে গেল। এটা তো তোমরা দেখেছো, এই প্রাইমারি আর সেকেন্ডারি ইমিউন রেসপন্সের ডিফারেন্স পরীক্ষায় আসে। অ্যান্টিজেন প্রেজেন্টেশনস, টি সেল কি করে অ্যান্টিজেন প্রেজেন্ট করে, একটা ছবি আছে, ওটা মনে রাখতে পারলে খুব ভালো হয়। দুটো ডিফারেন্ট পাথওয়ে আছে, একটা এন্ডোজেনাস আর একটা এক্সোজেনাস অ্যান্টিজেনকে ডিল করে, একটা MHC1 দিয়ে একটা MHC2 দিয়ে এক্সপ্রেস করে, টিসি সেলস আর TH সেলস রেসপন্স করে তার। এই সেই পাথওয়ে যেখানে এটা হচ্ছে এন্ডোজেনাস অ্যান্টিজেন, এন্ডোজেনাস অ্যান্টিজেন এখানে প্রসেসড হচ্ছে রেটিকুলামে, তারপর গলগি অ্যাপারাটাসের মধ্যে দিয়ে এটা এক্সপ্রেসড হচ্ছে ভায়া ক্লাস 1 MHC দিয়ে এক্সপ্রেসড হলো এন্ডোজেনাস অ্যান্টিজেন। এটা হচ্ছে এপিসি সেল, এটা একটা পুরো এপিসি সেল, তার মধ্যে দুটো ভাগ করে দুটো পাথওয়ে দেখানো হয়েছে, একটা এন্ডোজেনাস ভাইরাল অ্যান্টিজেনের জন্য বা ইন্টারসেলুলার অর্গানিজমসের জন্য, আর এটা এক্সট্রা সেলুলার অর্গানিজম এখান দিয়ে ঢুকছে, সে জাস্ট প্রেজেন্টেড হয়ে যাচ্ছে MHC2 দিয়ে, তার কোনো প্রসেসিং দরকার হচ্ছে না, সরাসরি এখানে এক্সপ্রেসড হচ্ছে। সে এই পাথওয়েটার ছবিটা মাথায় রেখো যে এটা হচ্ছে সিম্পলি এন্ডোজেনাসের জন্য আর এটা এক্সোজেনাসের জন্য। অত কিছু মাথায় রাখতে হবে না। প্রসেসিং হয় এন্ডোপ্লাজমিক রেটিকুলাম আর গলগি অ্যাপারাটাসের মধ্যে দিয়ে হয় MSC1, আর এখানে ডাইরেক্টলি প্রেজেন্টেড হয়। শুধু MSC1 আর MSC2 কোন ক্ষেত্রে প্রেজেন্টেড হয় সেটা মাথায় রাখতে হবে। নেক্সট, খুব সুন্দর ছবি। এখানে দেখো টি সেল, ম্যাক্রোফাজ সেল, তার মধ্যে ফ্যাগোসাইট ঢুকেছে। এই ফ্যাগোসাইট প্যাথোজেনটাকে নিয়েছে ওরা, নেওয়ার পর ওটা MSC ক্লাস 2 দিয়ে প্রেজেন্ট করেছে। এখানে এই পেপটাইড যেটাকে একটু প্রসেস করেছে দিয়ে পাঠিয়েছে। টি সেল রিসেপ্টর তাকে ধরেছে, ধরবার পর এই যে দেখো কো স্টিমুলেশন B7 থেকে CD28 এ গেছে। এখানে কো স্টিমুলেশন হয়ে ইন্টারলিউকিন 1 সিক্রিয়েটেড হয়েছে। এই যে B7, লাইগ্যান্ড B7 অ্যান্ড রিসেপ্টর CD28 একে লেগেছে। এই কো স্টিমুলেশন যদি না হয় তাহলে এই ইমিউন রেসপন্সটি হবে না। সুতরাং কো স্টিমুলেশন ইজ এসেনশিয়াল। তো এটা যখন হলো তখন টি সেল ক্লোরালি প্রলিফারেটেড হলো TH1 আর TH2 তে। TH1 সেল মিডিয়েটেড ইমিউনিটি আর TH2 হিউমারাল অ্যান্টিবডি, হিউমারাল মিডিয়েটেড অ্যান্টিবডির মধ্যে চলে গেল। এটা বি সেলের গল্প। বি সেলেরও এরকম সব মলিকুল রয়েছে। বি সেলের MSC ক্লাস 2 ধরবার ক্ষমতা আছে, IgM আছে। বি সেলস কি করে CD23 প্রসেস করে সেই গল্প আছে এখানে। CD3 কি করে প্রসেস করে CD4 কে, সেই সব গল্প আছে। সাইটোকাইন সেটা তোমাদের জন্য নয়। সিগন্যাল হলো ডিফারেন্সিয়েটেড হয়ে গেল, এখানে সেই গল্পই বলেছে। TH1 আর TH2 সাবসেটে ক্রুশিয়াল সাবসেট তৈরি হয়ে গেল, সেটা বলেছে। ইন্টারলিউকিন 2, ইন্টারফেরন গামা, এগুলো সব এখানে প্রডিউসড হচ্ছে। ইন্টারলিউকিন 2 এখানে পার্টিসিপেট করেছে ইন্টারলিউকিন 12 এর সাথে, তবে ম্যাক্রোফাজকে অ্যাক্টিভেট করতে পেরেছে, তবে নাইট্রাস অক্সাইড প্রডিউসড হয়েছে। এখানে অনেকগুলো আরো সাইটোকাইনস আছে, ইন্টারফেরন গামা, টিউমার নেক্রোসিং ফ্যাক্টর বিটা, এরাও পার্টিসিপেট করতে পারে। IL4, 5, 6 and 10, IL4, 5, 6 তো এখানে TH2টাকে ক্লোনাল পলিফারেশন করেছে, আর IL10 আর IL12 এই দুটোর মধ্যে একটা ইন্টাররিলেশন মেনটেন করে যে কোনটা কখন চলবে আর কখন কোনটা বন্ধ হবে। অনন্তকাল তো ইমিউন রেসপন্স চলতে পারে না, সেটাকে কন্ট্রোল করার জন্য IL10 আর IL12 কাজ করে। মেমারি সেলস তো সবকিছুরই আছে, মেমারি সেলস পরে করে। এখানে সেই ছবিটা আছে, যে ছবিটা এখানে আঁকা, তবে এই ছবিটা কমপ্লিকেটেড। এখানে দেখাচ্ছে যে অ্যাক্টিভেশন আর ডিফারেন্সিয়েশন অফ টিসি সেল, মানে টি সাইটোটক্সিক সেল বলে একটা সেল আছে, একটা গ্রুপ, তারা কি করে অ্যাক্টিভেটেড হয় সেটা দেখিয়েছে। এত মনে রাখার তোমাদের দরকার নেই। ন্যাচারাল কিলার সেলস, একরকম সেলস আছে যারা CD4, CD8 কোনোটাই এক্সপ্রেস করে না, তাদের বলে নাল সেলস। ওই নাল সেলের মধ্যে থেকেই একটা আছে, তাকে বলে হচ্ছে NKT সেলস। তারা সব বড় বড় দাদা, তারা যখন ইমিউন সিস্টেমটা খুব একটা কম্প্রোমাইজ কন্ডিশনে আসে তখন তারা ফিল্ডে নামে। NK সেলের গায়ে এক্সপ্রেসড হয় হচ্ছে এটা CD16 আর CD56, আর টি সাইটোটক্সিক সেলের CD3 আর CD8। এটা একটা CD8 সেলেরই একটা স্টেপ ফর্মুলা আর কি। এটার MSC রেস্ট্রিকশন নেই, যে কোনো সেলের এগেনস্টেই কাজ করতে পারে, যে কোনো অ্যান্টিজেনকে ধরতে পারে, কোনো MSC রেস্ট্রিকশন NK সেলের নেই। কিন্তু TC সেল MSC1 রেস্ট্রিক্টেড, আর এর কোনো মেমারি নেই, TC সেলের আছে। ADCCর কথা বললাম, ওই ADCC কি কি লাইটিক এজেন্টসের মধ্যে আছে। লাইটিক এনজাইমস, পারফোরিন, টিউমার নেক্রোসিং ফ্যাক্টর আর গ্র্যানজাইমস, এরা সবাই মিলে একসঙ্গে টার্গেট সেলকে অ্যাটাক করে। ওই ইওসিনোফিল, এখানে ইওসিনোফিলের গল্পটা এসছে। এই ইওসিনোফিল থেকে এইগুলো সব বেরিয়েছে, সমস্ত টক্সিক ম্যাটেরিয়ালস। নিউট্রোফিলস থেকে এসছে, NK সেলস থেকে এসছে, ম্যাক্রোফাজ সেলস থেকে এসছে, ম্যাক্রোফাজ সেলস থেকে এসছে। এই সব মিলিয়ে একসঙ্গে ইন্ট্রাসেলুলার কিলিংটা করতে পারে। অ্যাসেসমেন্ট অফ CMI আছে, সেটা কি করে বোঝা যায়? যে MLR, একটা মিক্সড লিম্ফোসাইটিক রিয়াকশনস বলে একটা জিনিস আছে, সেটা টেস্ট আছে আর কি, সেটা দিয়ে করা যায়। এটা আমরা ওই যখন কিডনি ডোনার তাদের মিস ম্যাচিং অফ দি অ্যান্টিজেন্স করি, তখন এই MLR রিয়াকশন টেস্ট করা হয়। Cell-mediated immunity is by virtue of NK cell, eosinophil, cytotoxic TC cell, all of them have. কারা কারা participate করে cell-mediated immunity তে? সবাই তো আছে এর মধ্যে NK cell করতে পারে, eosinophil আছে, এই যে এক ছবিতে রয়েছে eosinophil, CMI এ eosinophil হ্যান্ড ইন হ্যান্ড যেতেই পারে, cytotoxic T cell ও থাকতে পারে, সবাই মিলে করে। So all of the above. Perforins are produced by, লাস্টের ছবিটাই ছিল, যারা মনোযোগ নিয়ে দেখেছো তারা পেয়ে গেছো। Perforins, plasma cells, suppressor T cells, cytotoxic T cells, memory helper T cells, কারা করে? Cytotoxic T cells, right answer. Very good. Professional antigen presenting cells include all, ওই যে সেটা, dendritic cell, macrophage cell, fibroblast cells and B cells. K, fibroblast. Thank you.
Namely, means the agents which causes cancer or which have the potential to make a cell from normal to mutant cell and cause formation of neoplasia or cancer in colloquial terms. So the carcinogens are broadly classified into three varieties. Three group of carcinogens are there. Number one is physical carcinogens. Number two is chemical carcinogens. And number three is biological or mostly viral carcinogens. So in our class, we briefly describe today the process of carcinogenesis by these various group of agents. Now, mechanism of induction of tumor is called carcinogenesis, and agents which can induce the tumor or cancer is called the carcinogens. So, we can see this is normal cell, and carcinogen agent, it acts on normal cell and cause DNA damage, DNA injury, DNA breakage or mutation, chromosomal translocation, then propagation of carcinogenesis occurs by formation and subsequent replication and formation of cell lines of neoplastic cells. Now, chemical carcinogen, I told you. Second is physical carcinogen, and number three is biologic or viral carcinogen. So, the most important and most common is chemical carcinogens. Okay. So, chemical carcinogens are broadly classified into two types. One is directly acting carcinogens, another is indirectly acting carcinogens. So, it classified as initiators and promoters. The initiators, these are group of chemicals, they initiate the mutation. Okay. They, upon acting on the cell, the naive cell, they starts or they initiates the process of carcinogenesis. So they are called as initiator. And the second group is promoter. The promoters, they do not initiate the process of carcinogenesis, but they help synergistically after the action of initiators. They are called as promoters, and they helps to run the process of carcinogenesis from normal cell to cancer cells and their subsequent replications. So among the initiator, I told you, one is directly acting carcinogens, another is indirectly acting carcinogens. From the name, we can say that directly acting carcinogens, they are no need to metabolically active before acting on the cells. They are directly acting. So, among the directly acting carcinogens, they do not require any metabolic conversion to become carcinogen. But the indirectly acting carcinogens, these are chemical substance which needs to be metabolically active by certain enzymes in the liver. So, they, after the formation of inactive form to active form, and then they act as carcinogens. So among the directly acting carcinogens, these are most important: alkylating agent, acylating agent, okay? And the promoter, they have lacking the intrinsic carcinogen potential, but their application subsequent to initiator exposure help the initiated cell to proliferate further. So initiator doesn't require promoter for becoming a carcinogen, but the promoter must need initiators to act prior before it to become carcinogens. Okay, everybody? Now coming to the mechanism. So initiator carcinogens, from the name it induces the mutation, but promoter carcinogen is not mutagenic. Dose single for a short time, and it is repeated dose exposure for long time. Response towards initiator carcinogens is sudden, but promoter is slow. Change is permanent and irreversible for the initiator carcinogens, but in case of promoter carcinogens, they may be reversible when the noxious stimuli can be withdrawn, the process of carcinogenesis or the process of cell transformation may go backward to normal cell if the notorious stimuli or noxious stimuli has been withdrawn. Sequence: applied first initiator carcinogens, then followed by promoter. Promoter applied after prior exposure to initiator. So from the name, initiator initiates or induces the mutation, but promoter doesn't induces the mutation. It necessarily adds with initiator effect to become a carcinogen. Next, effective alone if exposed in large dose, not effective alone. I told you in several times, promoter carcinogens are not effective alone. Molecular changes, what are the molecular changes? Changes mostly include two very important genes for cancer development. One is proto-oncogene, another is anti-cancer gene. Proto-oncogene, that is RAS mutation, and anti-oncogene or anti-tumor gene, that is P53 or guardian of genome. So upregulation of the proto-oncogene and downregulation of the anti-oncogene or anti-tumor gene leads to growth and proliferation and replication of the mutated cell or tumor cell. Okay. Example: initiator carcinogens, most chemical carcinogen and radiation, and promoter carcinogen. These are short-acting estrol hormones, esters, etc. So I told you, among the direct-acting carcinogens, most important is alkylating agent, and second one is acylating agent. Alkylating agents: anti-cancer drug, cyclophosphamide, chlorambucil, epoxide, beta-propiolactone. Acetyling agent: acetylimidazole, dimethyl carbamyl chloride. So alkylating agent and acylating agent, these are direct-acting carcinogens. These do not require any metabolic conversion before becoming a criminal stimuli. Okay, they are directly acting. They directly acting on the genetic constitution or the DNA of the cell. Okay, epoxide normally found in the resin or varnish industry. Okay, so this is chemical carcinogen, and this is directly acting carcinogen. Anti-cancer drug, yes, anti-cancer drug. These anti-cancer drugs are also called cytotoxic drug. Okay, so anti-cancer drug normally designed to treat against one cancer, but conversely, some anti-cancer drug, as they are cytotoxic, as they are DNA modifying agent and they cause DNA injury, it may cause injury to the normal cell. Suppose one tumor cell, one anti-cancer drug is designated to battle or to fight against one group of cancer, but this is cytotoxic. The mode of action or mechanism of action of anti-cancer drug are broadly cytotoxic. They injure the DNA. They injure the DNA by making dimers or breakage in the double-stranded chains. This double-stranded chain breakage doesn't allow to proliferate cancer cell further, but conversely, it can cause DNA breakage and DNA damage to the normal cell of the other body parts as well. So one cancer drug, it may be beneficial to one group of tumor, it may act like a carcinogen to other group of tumor. Clear? So these are cytotoxic. Cytotoxic means the toxicity to the cell. So toxicity to the cell, to the cancer cell, is almost beneficial to us, but sometimes these anti-cancer drugs are attributed to cause cancer to other organs. Okay? Alkylating agents, I told you, they directly act on guanine base of the DNA molecule, and it causes breakage of the DNA strand, affecting the ability of the cancer cell to multiply. Bifunctional alkylating agent can cause intra-strand linking and cross-linking. So haphazard linking, A11T, C111G. This is the normal purine and pyrimidine configuration of the double helix DNA strand designated by Watson and Crick. But the anti-cancer drugs, or the alkylating agent, or the cytotoxic drug, or the directly acting alkylating agents, they cross-link the DNA strands haphazardly, they form dimer within the strand, and they form breakage between the DNA strands. Okay? Now, there are some indirectly acting carcinogens or pro-carcinogens. So, indirectly acting carcinogens or pro-carcinogens, they requires metabolic activation prior to become a carcinogen. Our common examples are coal, tar, soot, tobacco, okay, polycyclic carbon, industrial and atmospheric pollutants, liquid paraffin, coal, soot, okay, fossil fuel, tar. These are all the carcinogens. They are indirectly acting carcinogens. They need further activation by various enzymes, namely cytochrome P50 or monooxygenase, and they got activated in liver before becoming a potential acting carcinogens. So azodite, the people who are involved in coloring industry, paint industry, the people who are involved in making of varnish or ceiling color or any type of color, any kind of dye, they contain a azo group, and this azo group is our indirectly acting carcinogens, and it requires metabolic activation. Okay? Aflatoxin B1, it causes hepatocellular carcinoma. In your cirrhosis chapter, in your hepatobiliary chapter, you will find this toxin, naturally occurring products, alpha toxin B1, betel nuts or supari. These are also carcinogen, but indirectly acting carcinogens. It requires metabolic activation by cytochrome P450. Asbestos, arsenical compound, metals, heavy metals, not metal bands, metals like nickel, lead, cobalt, okay, insecticide, fungicides, aldrin, dieldrin, cordrin, saccharin, these are all indirectly acting carcinogens. They all require metabolic activation by liver. Promoter carcinogens, example are dietary fat in cancer of colon, endogenous or exogenous estrogen. Okay, dietary fat in cancer of colon, it mostly occurs in the developed country where the people ingest more smoked fish, more fat, more ham, more burger, more steak, beef steak or steak of red meat. They are more vulnerable to develop colon cancer due to increased amount of dietary fat. So, stages of chemical carcinogenesis, pathogenesis of chemical carcinogenesis, it requires several years to become a cancer. So, it works throughout the years or a long period of time. They act and they produce mutation in the cell line, normal cell line, and then the normal cell line got mutated, and the abnormal cell or mutant cell, they got replication, and a cancer cell line or neoplastic cell line is formed. And the factors that influence the induction of cancer are dose and mode of administration of the carcinogen, individual susceptibility, and various other predisposing factors. So, dose and mode of administration is very important. A small dose for a short period of time is insignificant than a strong sustained dose for more period of time, more long period of time. So, dose, mode of administration, and the time of dosing or the period of dosing or period of exposure to the particular carcinogen is very much important and important factors. Okay, so chemical carcinogenesis, it occurs to 3 lines: initiation of carcinogenesis, promotion of carcinogenesis, and progression of carcinogenesis. Mutation in the proto-oncogene and anti-oncogene. Proto-oncogene is RAS gene and anti-oncogene is P53, that is called also guardian of genome. Okay, so initiation of carcinogenesis, promotion of carcinogenesis, and then progression of carcinogenesis. First, initiation of carcinogenesis made by the initiator carcinogens, promotion by promoter carcinogenesis, and progression, that is the replication and proliferation of the mutated cell lines. So direct acting carcinogens are often called as electrophilic. Electrophils means love, affinity. So directly acting agents, they love electron. Why they love electron? Because they are deficient of electron. They are deficient of electron and they love electrons. So they are called electrophile. And these electrophiles, they directly acting to the DNA double helix structure and cause DNA damage. And indirect carcinogens, inactive form, it got activated in liver by the action of monooxygenase or cytochrome P450. Then active carcinogens, it also convert to electrophilic organs, electrophilic molecule, and then cause DNA damage. Okay, so metabolic activation, how the indirectly acting carcinogen, they got activated by cytochrome P450 or monooxygenase. It is a very important enzyme. It involves in very variety group of action, and it is a most important enzyme in liver of our human line. You will also listen this name of this enzyme in your pharma coaching or pharma classes. Okay, this enzyme, it after metabolism in liver, they become electron deficient and act as electrophiles. The pro-cancer carcinogens or promoter carcinogens, they are being activated metabolically by cytochrome P450, and reactive electrophiles bind to the electron-rich portion of other molecules of the cells such as DNA, RNA, and other proteins. So after metabolic activation, the indirectly acting carcinogens, they got deficient of electron, and whenever they are deficient of electron, they have developed affinity of electron, they are electrophiles, and the electron-rich portion of the cell like DNA, RNA, they directly act on the cell. Okay, so target of electrophiles is DNA producing mutagenesis, and containing damaged DNA is not required. So when the DNA injury or the DNA breakage or the DNA mutation is beyond the threshold of repair by the cell own homeostasis response, then the cell got mutated, and if the mutated cell, they enter into the cell cycle, into the from the S phase to G1 phase, then the cell, the mutated cell, they replicate by its own formula or own nature, and then cause the permanent and irreversible formation of mutated cell line. Okay, everybody clear up to this? So now directly acting carcinogens, these directly act as reactive reactophile and bind to the electron-rich portion of the other molecules of the cell such as DNA and RNA. RAS gene mutation and P53 gene mutation, very important. One is anti-oncogene, another is proto-oncogene. So up-regulation of the proto-oncogene, it induces the tumor, and down-regulation of anti-oncogene, it also causes the tumor. Promotion carcinogenesis, the promoter of carcinogenesis substance are phenols, hormones, artificial sweeteners, drug like phenobarbital. So this group of compounds or this group of chemical carcinogens, these are promoter carcinogens, and they require initiator carcinogens to act prior before their action, and they need metabolic activation, and this is the second stage. Now the third stage is progression. They do not produce sudden change. We see in your chart, that is the difference between the initiator carcinogen and promoter carcinogen, and the changes induces may be reversible. they do not damage the DNA per se and thus not mutagenic. So if promoter carcinogens are act alone, they cannot cause the DNA damage or DNA injury. But when they act after the initiator, they cause mutagenesis. Okay. So clonal proliferation and expansion of the mutated or initiated cell. So this is important. One, the DNA damage is there and the DNA damage is beyond repair. The cell is permanently damaged or the cell has the mutation within it. But this mutated cell doesn't help us if the cell doesn't proliferate or replicate by its own. If the mutated cell dies at this stage, it cannot be a tumor. Okay, the mutated cell must replicate or must proliferate. So the mutation that caused by the DNA damage must goes on subsequently in the following generations of the cell. Okay. Now progression of carcinogenesis is the third part. It shows the phenotypical characters of malignancy, morphology, biochemical composition and molecular features of malignancy. So the newly accumulated characterize, these cannot be there if the mutated cell doesn't replicate and if the mutated cell will die after the mutation. Okay, so the mutated cell must replicate before transforming into a neoplastic cell line. So this is the whole schematic diagram. Chemical carcinogen, it acts on normal cell. These are electrophiles. They cause DNA damage. It is the initiation. The mutated cell here acts the promoter. Then the cell division occur, and when the cell divides, okay, after the promoter carcinogens occur, when the cell divides, then mutated cell, it gives rise to cancer cell lines. So this is the initiation by the carcinogens, directly acting or indirectly acting. It acts on the normal cell. The mutated cell here acts the promoter. Then the cell division occur, and this is the most crucial part, that is cell division. If the cell doesn't divide, it doesn't give rise to the neoplastic cell line. Okay. So the cell replication, cell proliferation, and subsequent progression to the neoplastic cell line is called progression of carcinogenesis. So three main parts. Don't forget, biological carcinogens are mostly viral. Okay, the DNA virus or RNA virus, mostly virus. Okay, so biological carcinogens are viruses, parasites, fungi and bacteria. However, the role of virus in the causation of cancer is more significant. মানে 100টা বায়োলজিক্যাল যদি এজেন্ট থাকে তার মধ্যে 95টাই ভাইরাস। সো biological carcinogens are mostly virus unless told otherwise. Okay, therefore biologic carcinogenesis is likely viral carcinogenesis. So the virus are HPV, human papilloma virus, EBV, Epstein-Barr virus, adenovirus and HBV, hepatocellular virus. These are important. I will demonstrate you some large chart after it. Okay, bacteria Helicobacter pylori also causes GI or colon carcinoma, fungus like Aspergillus flavus, hepatocellular carcinoma, parasites like Schistosoma hematobium. But you may forget this bacteria name, you may forget this fungus name, and parasite name, but don't forget the virus names. Okay, these are the most important short note or most important question to be asked in your theory or in your practical or in your semester or in your final exams. So the viral carcinogens, the name of the virus, their genetic constitution, whether it is DNA virus or the RNA virus, and their causes of cancer, the name of the cancer caused by the viruses. Okay. So 20% of all cancer worldwide are due to persistent viral infection. Oncogenic virus are two types, like the viruses which contains DNA as their genetic constituents, like DNA virus, and which the viruses those contain RNA as their genetic constituent, they are called RNA virus. Very easy. Now this chart, Papova group of family, human papilloma virus, the most notorious criminal for the viral carcinogenesis and most widely spread virus. Okay, human papilloma virus. It can cause starting from simple skin wart, simple anogenital wart to the cervical carcinoma, vulval carcinoma, vaginal carcinoma. So it has variety of strains, it has variety of groups that causes the cervical cancer, anogenital wart, condyloma acuminata, multiple spectrum of diseases. But HPV variety are like 6, 1, 16, 18, 31, 33. Among this variety or these strains, HPV 6 and HPV 11 are mostly cause the genital wart, anogenital wart or skin papilloma, benign papilloma. But the strains, namely HPV 16, HPV 18, HPV 31, HPV 33, these are high risk HPV virus, and these are attributed to the formation of cervical carcinoma, mostly vulval carcinoma and vaginal carcinoma to some extent. But don't ever dare to forget HPV causes cervical carcinoma, and don't forget the strain which are the high risk strain of HPV, which are the low risk strain of HPV, which are the intermediate strain of HPV. Now next, herpes group of virus, Epstein-Barr virus and human herpes virus 8. Epstein-Barr virus, it causes Burkitt lymphoma, okay? This is a group of lymphoma, lymphoblastic lymphoma, L3 group of L3 type, Burkitt lymphoma. And human herpes virus 8, it causes Kaposi sarcoma. Kaposi sarcoma is a sarcoma. It also we can see in the patients of immunocompromised. Like in your microbiology classes, you will be taught that the opportunistic infection and the tumors of the HIV infected individuals. Then you must remember the Kaposi sarcoma, okay? So human herpes virus 8 initiated the Kaposi sarcoma mostly in immunocompromised individual, okay? Pox virus, this is not a cancer, this is molluscum contagiosum, is a viral wart, okay? Commonly occur in face, in trunk, near the perioral region. And hepatitis B virus, it is attributed for causing of hepatocellular carcinoma. Now, I told you earlier, cancer causing type HPV 16, HPV 18. Another is HPV 31, HPV 33. But most important is 16-18, HPV 16 and HPV 18. More than 75% of cervical cancer and more than 50% of vaginal and vulval cancer are attributed to this high risk group of human papilloma virus. Don't ever forget. And non-cancer causing types, HPV 6 and HPV 11. 90% of anogenital wart, benign skin papilloma, etc. Now diseases associated with Epstein-Barr virus, herpes group of family, Epstein-Barr virus, ki bolechilam? Burkitt lymphoma, very good. Burkitt lymphoma, তাহলে আর কি লিম্ফোমা হতে পারে? হজকিন লিম্ফোমা হতে পারে, বার্কিট লিম্ফোমা, nasopharyngeal cancer. Okay, the people who are working in a carpenter shop or the fume or the small minute particles of the wood industry, it also initiate after the action of Epstein-Barr virus. It also causes the nasopharyngeal carcinomas. Okay. Now another, these are the previous chart was DNA viruses, okay, the DNA viruses, and now this is chart of RNA oncogenic virus, and the most important is human T cell lymphotropic virus, okay, HTLV1 and HTLV2, very important, and it also causes the tumor or cancer in immunocompromised individual like Kaposi sarcoma, okay. So HTLV1, human T cell lymphotropic virus. So from the name, this RNA virus, we can say that this virus acts on the T cell of the system, and in AIDS or in HIV infected individual, this T cell, the cell mediated immunity is geoparadized. So the viruses which acts on the T cell, it causes adult T cell leukemia, lymphoma kind of cancer, and HTLV2 T cell variant of hairy cell leukemia. So all the liquid tumor. So the tumor which are mostly involves the blood and hematolymphoid systems, these are called liquid cancer, and the tumors which involves the organ like liver, like lungs, like brain, like bone. These are tumors called solid tumors, okay? And hepatitis C virus is also RNA virus, and it causes hepatocellular carcinoma like HBV. So this chart is very important in terms of exam, in terms of oral, in final exam, or in your semester exam. The viral oncogenesis and the name of the viruses and name of the tumor that caused by the viruses, okay? So viral carcinogenesis or viral oncogenesis, mode of DNA viral oncogenesis and mode of RNA viral oncogenesis mostly similar, okay? Replication and integration. If the virus infect the host cell and introduces into the genetic constituents of the host cell, if the virion occurs or virion produces, then the process is called replication. So replication and virion formation is not tumorogenic, or this is the viral infection sequel, but this is not tumorogenic. But when the genetic constituents, that is the DNA and the RNA constituents of the viruses, integrated into the host DNA and cause DNA or RNA damage of the cell, then there is mutation and there is tumorogenesis, okay? Amader viral infection hoy, commonly viral infection hoy, shobai sob viral infection theke tumor hoy na, karon segulo viral replication hoy, virions hoy. কিন্তু যদি ভাইরাল ইনফেকশন সাস্টেইনড হতে থাকে, প্রলং পিরিয়ড ধরে হতে থাকে, সেক্ষেত্রে কখনো the virus gets integrated into the host DNA and then causing mutation, and the mutated DNA after replication then causes mutant cell line of the virus infected cell or the tumor cell, okay? So if there is replication, the virus may replicate in the host cell with consequent lysis and infected cell and release of virion. That is no neoplastic transformation and death of the cell, okay? This occurs, and this is open and shut case. But integration, when the viral DNA may integrate into the host cell DNA, then neoplastic transformation by inducing mutation into the DNA. So for becoming viral carcinogenesis or viral carcinogen, the DNA or RNA or the genetic constituents of the viruses needs to be, or obligatory needs to be, integrated within the host DNA to become a cancerous cell. So here is the picture, okay? So this left hand side show replication and release of virions, but whenever there is the viral DNA integration within the cell, there is no release of virion, but here is the mutation of the host cell DNA, okay? And this is called integration, and this integrated cell or transformed cell gives rise to the tumor cell line or cancer cell line, okay? Mode of RNA viral oncogenesis is same, the integration is same, but the enzyme that is present in DNA virus is DNA polymerase, but the enzymes in RNA viruses, it causes what? Reverse transcriptase. So reverse transcriptase is the enzyme that makes complementary DNA strands from RNA strands. So reverse transcriptase, okay? So after action of reverse transcriptase, these viruses forms the complementary DNA strands to the, that is complementary to the host DNA, okay? Now this is viral RNA, and after entering into the cytoplasm, DNA synthetase or reverse transcriptase, that is viral RNA to viral DNA occurs. So from viral RNA, this is the RNA virus, after action of reverse transcriptase, this viral RNA, it forms the viral DNA or provirus. This viral DNA or provirus is integrated, okay? Integrated into the host DNA, and this integrated genomic sequence or altered genomic sequence or the mutated DNA sequence, whenever they replicate and form the messenger mRNA, and then viral protein occurs, and this is the malignant cell line or mutant cell line, okay? So viral oncogenesis, the main important thing, when the virus replicates and cause cell deaths by release of virion, that is not tumorogenic, but when the viral DNA or RNA or viral genetic material is integrated into the host DNA, then there is the integration and mutation of the host DNA cell line occur and formation of the cancer cell line occurs. Now, viral RNA genome as well as reverse transcriptase released into the cytosol, double-stranded viral DNA or provirus, I told you earlier. Integration, induce mutation, thus transform the cell into neoplastic cell. Now, last, the physical carcinogenesis and the carcinogens and carcinogenesis. Mostly the radiation. The radiation of the physical carcinogens are ionizing radiation, UV rays, nuclear bomb, radium. Okay, these are the physical carcinogens. Ionizing radiation, the most important. It ionizes the host DNA. It directly acts the host DNA for breaking, for intra-dimer formation and intra-link stranding. Okay, radiation and non-radiation physical agent. The radiation physical agent or the radioactive physical agents are most important among the physical carcinogens. Okay, radiation physical agents are mostly ultraviolet light, UV light and ionizing radiation. Okay, this is UV light. Dhalai korche. Okay, welder arcs. These are also carcinogen. Long standing, sustained. Okay, exposure. It causes DNA damage. UV light, it causes DNA damage whenever the protective melanin layer is breached or the melanin layer is less. Jaunty roads. Okay, হয়েছিল তো skin cancer, melanoma. তাহলে the whites are mostly susceptible, are developing skin cancer or tumor that are caused by the ionizing radiation or UV ray radiation. Now, how the UV ray of sun occurs? By breaching the stratosphere and the other ozone layer mostly. Okay, the greenhouse effect ozone layer, it is breached by our civilization. Then this breaching ozone layer allows the UVA, UVB and UVC with their respective wavelengths. It reaches on Earth and it acts as physical carcinogens to the people of mostly near the people who lives near the equator. As the equator region people has the highest chance to get maximum volume of light or maximum lux, 11-12 knowledge. Okay, and in humans excessive exposure to UV rays can cause various forms of skin cancer like squamous cell carcinoma, basal cell carcinoma and malignant melanoma. Now there are few pictures. Fair skinned Europeans are more susceptible. Albinos who do not tan readily, albinos means they readily lack the melanin concentration in their skin, and melanin has a more powerful protective action against the harmful UV radiation or any ionizing radiation of our environment. Inhabitants of Australia and New Zealand, okay, living closer to the equator, who receive more sunlight, farmers and outdoor workers due to effect of actinic light radiation. So more sun exposure, more outdoor activity, less melanin, albinos are more susceptible to develop cancers like squamous cell carcinoma, malignant melanoma, basal cell carcinoma due to action of ionizing radiation or UV ray from the sun. So mechanism of carcinogenesis. Carcinogenesis is, so it enters the skin by breaching the epidermis, and whenever the melanin or protective melanin layer is deficient, then it causes induction of mutation by DNA damage, directly acting physical agent, DNA damage, making cross-links, making intra-link strands, making dimers, haphazard arrangement beyond the double helix structure, beyond conventional double helix structure. Others mechanism are inhibition of cell division, inactivation of enzyme and cell death. But the most important mechanism of action of UV radiation is induction of mutation. How the induction of mutation occurs? like two consecutive bases on one strand to bind together. So it acts on pyrimidine dimer in DNA. So incoming UV photon, it breaks the DNA double helix structure A double bond T, C111G structure, and it causes the intralink strand. Instead of making strand between the two strands, this UV radiation makes strands or make base formation within a single strand, and this is the mutation, and this is causes the cancer. Okay. Again, oncogenes, in particular rash oncogene and anti-oncogene. So coming to the ionizing radiation, X-ray, alpha ray, beta ray, alpha ray by uranium, radium, beta ray emitted by decaying nuclear fuel, gamma rays, proton, neutron. So these are ionizing radiation. It damages the DNA strand by ionizing or remove electron from one atom. Okay, ionizing radiation. All forms of leukemia except CLL are caused by the ionizing radiation. Other are cancers like thyroid, most commonly papillary carcinoma of thyroid. It has good prognosis, but this papillary carcinoma is mostly attributed to the ionizing radiation. Like X-ray, like alpha ray, like gamma ray. Okay, X-ray, it is also beneficial, but repeated exposure to X-ray is not beneficial to us. So repeated X-ray exposure or repeated X-ray exposure to neck area, it causes papillary carcinoma of thyroid. So minors of radioactive elements have higher incidence of cancer. So the miners in America or in other developed country who has got their uranium stored in their land, so the miners who work upon radioactive organic material, these are higher susceptible to develop cancer. You know incidents of Chernobyl, a very good series in Netflix. Chernobyl, there is a city in Ukraine where there is nuclear reactor factory was there, but due to accidental leakage, okay, in the year of 1985, the entire population in that city of Chernobyl wiped away, okay, and the survivors in that city are developing cancer and still they are affected by the cancer. So this is a city of historical importance, and this city has been abandoned. There is a very good documentary in Netflix, in our other OTT, Chernobyl, you may say, okay, you may see. So there is a nuclear reactor factory in Chernobyl. The similar incidents, it occurs where? In India, anybody? Huh? Bhopal, very good, Bhopal, in the year of 1984. Okay? Bhopal, etao to series hoyeche. Huh? K.K. Menon, ki naam? Railway Man, very good series. Okay, tahole otao ekই জিনিস. The entire city wiped out in overnight, and the survivor, they are not safe as of present day. They are more prone to develop cancers. The survivors of Hiroshima and Nagasaki, the fat boy and the little man, this is the biohazard or atomic hazard emblem. Okay, toxic, radioactive, toxic. Japanese atom bomb survivor, they are more prone to develop malignant tumors, both solid tumor and liquid tumor. Liquid tumor like acute and chronic myeloid leukemia, and various solid tumor of breast, colon and thyroid and lung. So the survivors and the progeny of the survivors are more susceptible to develop cancer. Why? Because the DNA damage, the mutation, the DNA breakage, the altered DNA or altered genetic constitution, they runs in their cell line, they runs in their family line. Okay, so they are more prone to develop cancers. High incidence of osteosarcoma in the people or the ladies who has fine fingers and are mostly employed in the radium watch factory, okay? Watch factory, they used to polish the numbers and digits and the hands of the watch by radium, and they brush their radium digits with their mouth, okay? So radium gulls, they are also called American watching gulls. Radium gulls engage in painting the dials with luminous radium, and this is radium, this is also radioactive, who unknowingly ingested radium while using leaves to point their brushes. The high incidence of osteosarcoma occurs in there. These are all historical things you need to know, but may not be asked in your exam. This is for your interest. So mechanism of carcinogenesis by ionizing radiation, it may directly alter the cellular DNA, or may ionizing, like it may dislodge ion from water and other molecules of the cell and result in highly reactive oxygen species or highly reactive free radicals. So either they damage the DNA by directly, or they produce free radicals that causes cancer in long term. Indirect route, radiation, ionizing water, formation of free radicals and damage of DNA. And direct route, it directly cause DNA damage like breaking, like formation of pyrimidine dimer and cross-linking, okay? Either indirect route or direct route. But the damage of DNA is most important. It may cause chromosomal breakage, translocation or point mutation. So it also depends on various factors like chemical carcinogen, type of radiation, dose and dose rate, frequency, and various host factors such as age, individual susceptibility, immune competence, hormonal influence, and types of cells irradiated. These are the factors. Non-radiation physical agents, mechanical injury to the tissue, or prolonged contact with certain physical agents like Kangri cancer, ekhane chobi nei, Kangri long-standing ulcer of the hand or extremity, it may subsequently give rise to squamous cell carcinoma. Kangri cancer is a type of cancer used, it commonly seen in the region of, anybody? Kashmir, very good, Kashmir. Kangri is a device of burning chula or burning coal, and the people of Kashmir, as they are very much devastated by the never-ending winter, they used to put the kangri within their abdomen or close to the abdomen, close to the core body. Then for repeated exposure and repeated prolonged exposure, they develop carcinoma of GI tract. This is also called Kangri cancer, okay? So, like this is, Kangri is a physical agent and it involves the repeated exposure, prolonged exposure, repeated contact, and it also called the long-standing cancer. Asbestos, the people who worked in a factory of asbestos, they are commonly develop malignant pleural mesothelioma and other tumor, asbestos adenocarcinoma of the lung, but most important is malignant mesothelioma of pleura, okay? So asbestos is a notorious physical agent and physical carcinogen. Workers engaged in hardwood cutting, I told you earlier, hardwood cutting or engraving having high incidence of adenocarcinoma of paranasal sinus. Cut guro kore, cut jokon সমান করে গুঁড়ো ওড়ে তো ওই গুঁড়োগুলো নাক দিয়ে ঢোকে, 1 বছর 2 বছরে কিছু হবে না, কিন্তু 20 বছর 30 বছর বাদে ওই গুঁড়োগুলো থেকে জমতে জমতে কার্সিনোজেনেসিস তৈরি হবে। অ্যাডিনোভাইরাস যদি থাকে তাহলে ন্যাসোফ্যারেঞ্জিয়াল কার্সিনোমা, অ্যাডিনোকার্সিনোমা অফ দ্য প্যারানেসাল সাইনাসে, যেটা এপস্টেইন-বার ভাইরাস হয়, এপস্টেইন-বার ভাইরাস দিয়ে হয়, সেটা এদেরও হতে পারে, এরা মোস্ট কমন। সব ওয়ার্কার দিয়ে হবে তা নয়, তার মানে ফ্যাক্টর হচ্ছে ইন্ডিভিজুয়াল ইমিউনিটি লেভেল, age, types of cell irradiated, time of exposure, mode of exposure, etc. Thank you everybody.